Alright everyone, welcome to my lab. So for this week, we are going to talk about how to use some bioinformatics tools to analyze the ChIP-seq data. A few announcements before our lab. So for this week, I wouldn't hold the audition office hour because of the spring recess, and we would hope you guys all have a very nice break. So the homework for is just due the first weekend after your break, so just take your time. And we got a lot of things to cover for this week. So to begin with, we are going to talk about the Odyssey Fest session. We are going to talk about how to manipulate the available modules on the Odyssey, and I also would provide you guys some examples and also practices. And then the main part is about how to use the Max2 package, which is developed by Dr. Liu and her lab to analyze the ChIP-seq data. And then I also made some summary about some very useful Bedtools commands for you guys for your references, and also some instructions about how to use the UCSC Genome Browser to visualize your ChIP-seq data analysis results. And at the last, I would take you guys to go through all the homework questions one by one. So to begin with, for this week, the Odyssey session, we are going to talk about how to manipulate the available modules on the Odyssey. So the question is, actually, when you are trying to do your research, you might want, the first step is just to check whether you find some really useful packages that you want to, you might consider to be used in your research, but you are not quite sure whether they are available on the Odyssey. So how could we check whether your target module has already been installed on Odyssey? Yeah, if it is not installed, you have to ask the FastRC friends for help. So before we do so, you have to log into your Odyssey account, and you should you are suggested, highly suggested, to run this command as the SRun to apply for an interactive session, so that you could it's best for all of us. And when you I have already applied one, so when you log into your home address, and how could we check whether our target module is available on Odyssey? For example, if you want to check whether Python is available on Odyssey, you could just type into module spider Python. Okay, great. So after this command, it tells you all available versions of Python on the Odyssey system. So if you want to use the most updated one as the Python version 3.6.3, you can just type it again as with the module spider command. Yeah, then it would tells you whether there are some pre-requirements for loading this target module. So for this one, it could be loaded directly. So when you are trying to do so, you just module load Python 3.6.3 followed by the nodes, and that stores the original package. But for this homework, we are mainly going to use the Max2 package. So you might consider to check whether Max2 is available on the Odyssey system. So remember our command as module spider max2. Alright, so it tells you all available versions. There are 4 of them, and if you want the most updated one and want to check whether there are some pre-requirements for loading this module, just type module spider, the most updated version of our Max2 package again. And after that, you would get the pre-requirements as this module called CentOS 6 have to be loaded first before you load Max2. So if you want to load, Max2 with version 2.1.2, you have to load the CentOS 6 module first, which is the answer we listed here as module load CentOS 6, and then load the module load Max2. Alright, now it's some practice time. So if you want to use the first question is asking you guys to use the BWA to do some mapping work. So if you want to check whether BWA is available on Odyssey, what could you do? Just remember our command as module spider BWA. Okay, so we got 5 versions of the BWA on Odyssey, and if you want to check whether there are some pre-requirements before you load the BWA module, you just type in module spider BWA. Yeah, it tells you that this module could be loaded directly, so you don't need other preload issues. And then it's about another example is the SAM-tools. Well, SAM-tools is not a requirement for this homework, I have to say, but I found it very useful when doing some data manipulations for the BAM file and the BET file. So I remember for one of the question, I think it's question 2 or 3, it's asking you to calculate the successfully mapping rates proportions and also the race proportions of the unique ones. So if you want to do so, you could SAM tools might be your friends. And if you want to load this module, how could you check whether they are available on Odyssey? Again, it's module spider SAM tools. Yes, we got a lot of versions of the SAM tools, and if you want to use the most updated one, and then want to check whether there are some pre-requirements for that, what could we do again? So it's module spider with the most updated version of the SAM tools. And then it tells you that it could be loaded directly, so it's so sweet, and you are all set to go. Great, that's the main part of our Odyssey Fest for today, and then this is the main guide for our lab material today. So it's about the ChIP-seq analysis. Well, ChIP-seq technology is actually popular to identify binding sites of transcription factors and other DNA binding proteins. It's very popular in around 2013 and 2014 since it requires less starting material, it costs lower, and also have higher peak resolution than ChIP-seq. But yeah, it uses the BAM files to align ChIP-seq data. So if you want to check the actual data format of the BAM file, you can go to the UCSC's Genome Browser website. It covers all the details about all very classical biological sequencing data format. For next week or so, we will touch the MAF format and also the VCF format, which are quite useful for the WGS data, which is the whole genome sequencing data, and also for the analysis of the statistical genetics. So, but these two files are not very good and do have some disadvantages. And our lab is actually currently actively trying to develop a new data format called the GDS to do some reservation for this old format. So if you guys are interested in that, I will touch that in the next lab also. Alright, sweet, let's go back. So this is the brief summary for the ChIP-seq protocols. You can check the original paper for details. So when it is popular in 2014 and 2016, there are even some really large-scale in-dream challenges to ask the scientists across the whole country to collaborate together to make some prediction on the in vivo transcription factors binding sites across the whole human genome. So actually, I get participated in this project in 2016. So even though it is a closed project, but it do provide us a lot of useful resources. I attached all the links to this original website here. For example, if you go to the original website, you can find their databases here. They have some really good database resources. So for example, this is the TF's motif database. I think one of the question is question 8 or it's question 8. You might consider using one of the tools listed here. So for example, one of the suggestions is the Homer database. You can check the database from this link. And also a lot of other interesting things. Just explore them by yourself if you want to. And then I also find a very interesting MIT open course. So it covers all the details about the ChIP-seq protocols. I also attached the links on the our Canvas website in the video session, which is a good video session on the discussion board. Okay, so even though ChIP-seq technology do have some advantages, there are also some difficulties. So there might be two main biases. The first one might be due to the sequencing and mapping biases. So Dr. Liu's protocol has solved, which is the MAX2, as you know, have solved is mainly dealing with this problem. And there are still another problem remained. So it's the chromatin structure issue. So you can imagine that when the chromatin are overlapped with each other in the 3 dimension, so it might be hard to do the ChIP-seq experiment to separate each of them. But uh, what would we solve this problem? For me, I might consider that using the 3D genome analysis procedure to deal with this topic. Well, uh, for example, if you for example, if you want to, for me, my main work is about doing the GWAS study and also how to improve the power of a GWAS by incorporating the information of the transcription factors binding site and also other epigenetic factors. So if you can imagine that these factors might influence each other in 3 dimensions because of the 3 dimensional chromatin structure. So if we want to solve these problems, we might need some data which could measure the three dimension of the genome structure. Well, actually, we got some help from the broad scientists. So this is a very interesting video talked by the professor from Broad Institute.
I'll work on modeling chromosomes. That's actually, can anybody guess what this picture is? Some medical students told me it's a karyotype, but if you zoom in, these are socks.
Yeah.
It's an excellent exhibit in London in the Wellcome collection.
Well, actually, it's trying to discuss the modeling of chromosome organization. Yeah, you know that the professor used the socks to model the chromosome, but I think you guys all have your own model to model the chromosome structure. But the basic idea is to model them in 3 dimensional way, and you can check out this video about how they could do so, and also another video which covers about the details about the sequencing technologies.
Usually I'm a professor of physics.
About the high-C data and which could measure the genome in 3D way. Alright, then yeah, later of the biases caused by the chromatin structure. Then we would focus on how to address the biases caused by the distance shift. So this is dealt with the Dr. Liu's paper about the max. And actually, so Dr. Liu just finds that the text might shift it towards the 3' direction, and we have to measure the distance and control these biases with shifting all the text by half of the distances. But another really interesting problem addressed by this paper is that the peaks that you are calling may not be as rich as your motif. So if you only need to do the typically people are using the lambda. So the lambda, the global lambda, to measure the Poisson distribution of the text in the global view. But if you only do not use the local lambda and the text shifting, you might got a lot of biases. So for this software, it cuts the genome into very small windows and estimate the local lambda for each window, and then do some summary analysis about the local lambda to improve the accuracy about the peaks. Alright, then for our lab, the main part is about the pipeline. So to begin with, we have to do some alignment of the original rates of the ChIP-seq experiment. Well, the original rates are actually stored as the FASTQ file. So this is what the first question is doing. You have to align these FASTQ rates into some output as the BAM files. So for example, I could lead you guys to check out this data structure. If you could go to our lab for our course folder. Yeah, there is a FASTQ file which is a proportion from a very large FASTQ file. So we prepared it here just for you guys to do the first homework to get an experience about how the mapping procedure is working. So the two BAM files are actually the result of the alignment, and they are needed for the following analysis. So after you do the alignment now, you could do some quality control on the alignment result, and then so the quality control stuff is about how to remove the duplicates. Actually, it inputs the BAM file and with the bed files as the output, and then this is a down sample procedure. So if you can check the original data structure, you might find that the sample size in the control file and the sample size in the treated files may not be equal to each other. Well, researchers suggested that such kind of issues might also introduce some mapping biases. So they want to do some randomly sampling procedure to random sample some samples from both control and treated ones. And then after doing the quality control, we could use the main part called the ChIP-seq to input the BAM file and with the bed file as the output, and then visualize all the peaks by using the UCSC Genome Browser or other things such or other things. So then for the quality control, it's about how to remove the duplicates. Well, it's very simple with the command and the filtered up. So -i followed by the input file and -g followed by the genome size. Well, --keep .dup is the default setting with the duplicates to keep. So for now, we don't want more duplicates, so we just use one here. Well, the default setting is also one, so you don't have to change that threshold. And then the -o followed by the output file. So, okay, so now let's go back. We have to check how could we load this module again. Before doing so, you have to do check whether max2 is available on Odyssey. So remember our command as the max module spider max2, and it tells you that a lot of available versions, so you can check the most updated one for the pre-requirements. So you have to load the first one before you load the max2 module. Okay, coming back. After doing the quality control, we are now going to do some downstream analysis. Oh well, this is not for this homework, but just provided here for references. If you want to randomly sample some samples from both the controlled one and the treated one to keep their samples as equal to each other, you can use the downsample command, which is the resident sample in the max2 package. Then it's the main part of this package is called the peak calling. So with the peak call, the -t followed by the treatment file, -c followed by the control file, and also -f followed by the file format, and -n is the output prefix, and -g is the genome size. Alright, sweet, we have already finished the max2 procedure, and then it's the bed files format. So you as you see, we would obtain a lot of bed files in this procedure, and I would say that bed files is the most easiest one for analyzing all the sequencing data. It actually just requires 3 required fields for the data structure, which is the chromosome and the start position and also the end position. Then you could you could include as much annotation scores as you want. So the Bedtools are actually really friends for some AP guys because they are generating a lot of epigenetic function annotation factors each day, and they really enjoying this format. So you can also go to the UCSC's Genome Browser to check out the details about the bed formats here. It covers every detail. Alright, let's go back. So I made some really useful Bedtools for your references here. For example, the sort command. If you don't know the function of each of these commands, you could just go to the Google and type into this command. So this tells you how does this command work. For example, if your original bit file are not ordered by the actual order of the chromosome and its position, after the sorted command, it could be ordered according to their chromosome and their positions. And also other very useful command. Feel free to check them out on the Google. And then the final part is to use the UCSC Genome Browser to visualize your results. So remember that we have already completed the alignment procedure and also the quality control, and if necessary, you might consider doing the random sampling procedure, and then counting all the peaks and storing all the peaks into a bit file. And now we might want to visualize the bit files. So how could we visualize the bit files about containing all the peaks we have for now? For me, I have already done some analysis this afternoon before our lab. So I have already completed all these 4 steps and get the bit files about the peaks of our data. I just want to show you guys how does this data look like. So if you can check to the alright, so for this file, so this bit files is actually the peaks file. Oh, this is the peaks file. So if you can go to the peaks Excel file, you can see all the details about the threshold that you are using. So this is actually the fault change. So if you want to change the fault change, you could change the threshold here as the -fe -cutoff 5 here. So I think for question 6, it's asking you to change it from 5 to 6. Oh, it's for question 5. So you can just check the parameter, change the parameter here. Another parameter for changing the for FDR is the -m command. It should be followed by 2 valid numbers, which is the low threshold and the other one for the high threshold. You can see that the -g is actually the genome size. So what we are using is the human genome. So you can type HS here or type the actual size of the genome here. Both are fine. Then after doing so, you might get a bit file, and if you can check the valid line of the bit files, it actually gives you the successfully mapped peaks here, number of peaks here, and if you can visualize the position of all the peaks, you could go to the details about them. So if we want to visualize them on the UCSC's Genome Browser, go to the website, select the human species, and type in the actual position. For example, if we want to begin with this guy, to let's see, this guy, just type into the position, and then you got the whole result. So the most important biological part is this one, and if you click this gene, it will give you all the annotation details and all its functions and related data resources. Yeah, you can play around with it. Alright, sweet. Then we can go through all the questions one by one if you are willing to join me to go through them. So yeah, the homework and also the lab materials are uploaded on our GitHub. So the first question is asking you to do the BWA, use the BWA to do some original data alignment. So you got the FASTQ file as the original ChIP-seq data, and then after doing the BWA alignment, you might got some SAM files and also some BAM files. So for here, it also asks you to calculate the uniquely mapped proportion which might be quite tricky if you have to do it, calculating them by your head. So for here, I provide the suggestions about using the SAM tools to do so. So if you want to use the SAM tools, you have to load the SAM tools, right? So the SAM tools could calculate the proportion directly. So if you want to use the SAM tools, you have to load it first, right? So how could we check whether SAM tools is available on Odyssey? Again, it's our command as module spider followed by the name of your tools. Whereas we got a lot of SAM tools versions, and what if we want to use the most updated one and to check whether it has some pre-requirements? We just need module spider the most updated version. So it tells you that it could be loaded directly. So you could just module load SAM tools what, and then on this interactive node, if you want to calculate the successfully mapped proportions, we could just type in the SAM tools flag stat followed by the output of your first mission, which is the result of the BWA. Oh, sorry, I didn't load the module first. So we have the module load the SAM tools. Oh, sorry, there is a mistake here. It shouldn't be a dash, it should be a slash. So we might encounter some error at this part. Let's see. You know what? Just forgot it. We could open another session. Oh, great. It already has the result. So we have to change the dash into slash. Just be careful about them in your homework. Great. Now we have to do so. So we want to calculate the successfully mapped proportion. So SAM tools have already provided this techniques. You don't have to calculate it by hand. So from the result, you can see that around 97%, 96% of the original rates have been mapped successfully. And if you want to calculate the unique mapped proportion, you could actually use this command to pick all the unique mapped peaks, unique mapped rates into the unique BAM file, and then use the SAM flag statistic summarizer again. At this moment, it gives you the total number that is uniquely mapped. So you could use the number divided by the number in your first mapped file to see the percentage. If I didn't remember wrong, it should be 85%. Great, then it's for question 2. It's asking you to do the quality control. So what you need is all the commands in the lab material. And for question 3, it's asking you to do the main part of the peak call. So just use the copy command. But for this question, it's asking you to do the copy command on the control and the treated examples separately. So it's using the lambda of the background by its default setting of the Max2 software. So for this part, the input file would only be the treated examples, which is the 1-bit file. You wouldn't need the control example for here. So you have to, for each one of the procedure, you would only need one bit file as the input data, not 2 of them. So do them separately for both the treated example and the control one. Then for question 4, it's now asking you to compare the control example and the treated example. So now you need 2 inputs. The dash T is the treated the bed file, and the -c is the bed file for the control example, and then followed by all the threshold and parameters. Oh, by the way, how could we change the for FDR? So you can change the FDR with the parameter followed by the -q, and you can also change the fold of the change by change the parameter here followed by -fe cutoff. So I can give you guys a intuitive understanding of what I'm talking about. If you go to the co-peak result, you can see that I got some Excel file. Yeah. Oh, now you can see that the meaning of all the parameters. So the effective genome size, which is the human species, is around this number. And then the fold change, it gives you the lower threshold and also the upper threshold, and also the cutoff we are using for the FDR as the 0.05. So the additional change, you could change it to 6 here. So as just change the -f e cutoff from 5 to 6. And I think for this question, is it asking you to? Oh, so for the question 3, it's asking you to estimate the fragment size in each of them. So which is the actually fragment size? It's this number actually, the distances as the about 159. So it varies across different max2 results. Just pay attention to that. Alright, then after doing the first part, the remaining part are all about the website searching. So you have to learn some website tools by yourselves. So the Cistrome database is actually provided by Shirley's lab. So the first question might be very simple. It's if I didn't remember wrong, it's asking you to check the post-it. Yes, it's asking you to calculate some prostate, to deal with prostate. So just prostate, and type in also the transcription factor as AR. Then you could do search. It gives you all the results related to them. So you can see that there are actually 20 results about that. So for this question, are there over 10 AR ChIP-seq data available in human prostate tissues? Yes, there are definitely more than 10. So that's all for question 5. And for all the remaining part, I think you can go through the Cistrome database and also the website to check them out. Alright, good luck with your homework, and really thank you for joining me in this lab. And again, hope you guys are enjoying your spring break. If you find any problem, please shoot me an email, and you can also check me out on the office hours in this week and also the week after our spring recess. Thank you guys, and good luck.